Alright, welcome back. In this video, we're going to go over enzyme inhibition. I apologize if I sound a little different today. It's because I am recording this using my Sony headphones outside my hotel. So I am on vacation for Christmas break, and I'm over here in Florida, and I didn't bring my whole mic set and whatnot. So I'm recording this using my headphones. So I'm on my balcony in my hotel room, looking at the night sky, recording this video because the grind never stops. So let's do this. So enzymes can be inhibited to alter enzyme kinetics. The three different types of inhibitions is competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. This is a very high yield topic on the MCAT. The MCAT loves to talk about these three things. So that's where we're going to go over it. So here's the little agenda. We're going to go over each of the types of inhibitions. We're going to go over how it affects Km and Vmax. And then we're going to look at the Lineweaver-Burk plot. So let's do this. The first type of inhibition is called competitive inhibition. So this green thing is the enzyme, the yellow thing is the substrate, and the pink thing is the inhibitory molecule. What competitive inhibition is first, this substrate wants to bind to the active site of an enzyme. That's how we get this enzyme substrate complex, to be formed to create a product. So the substrate must bind to this active site to actually get a product. Well, what's happening is this competitive inhibitor is binding to the active site where the substrate should bind to. So it's basically in the way. So the substrate is not able to actually bind to the enzyme because it's being blocked by this inhibitory molecule. This is what competitive inhibition is. Now, here's the interesting thing, is that if you were to have enough substrate, it'll actually kick off this inhibition molecule, and we can proceed as normal and create product. So let's relate this to a real life scenario. Not relating enzymes, but I want to teach it like elementary school. So let's say this. Say the enzyme is a toaster. The substrate is a piece of bread. And say the competitive inhibitor is like a burnt toast that's left over, like crumbs in the toaster. Okay, so the toaster has 6 available spots for pieces of bread. The substrate, say we have 3 pieces of bread that's inside the toaster already. We put 3 pieces of bread inside. Now the other 3 spots are basically being blocked by like burnt crumbs of toast from previous stuff. Just say. So the bread, the 3 fresh slices of bread cannot actually be, get in there. It's being blocked by burnt crumbs, basically. The toaster's dirty. Well, this is what we can do. If we take a bag of bread, just say like 500 pieces of bread, and we just pour it over the toaster, there's a high chance that there's going to be at least 3 slices that are going to fit inside that basically blocked hole because of the burnt crumbs. In other words, it's going to kick out the crumbs. It's going to basically wedge its way in that hole because we just have so much, we have so much bread that there's not enough space, so it basically just shoves its way inside the toaster and kind of like basically, moves the crumbs out of the way. It's such a dumb example, but, I can't really think of anything else. So what basically is happening is that if you have a lot of substrate, it just basically kicks off this inhibitory molecule and we go back to normal. So let's look at the Lineweaver-Burk plot. The yellow line is the non-inhibitor, so basically absolutely normal, like the wild type. The inhibitor is in pink. Notice that we have a slope change. We get steeper, but the y-intercept is the same. So what's happening is the Km increases. Km increasing is the same thing as saying, lower binding affinity. Okay, it's a lower binding affinity. Because let's think about this. Let's go back to the toaster again. So if The toaster has 6 spots, but 3 of them are being blocked. That means only 3 pieces of bread can bind. That's a lower binding affinity than compared to the natural 6 pieces of bread if nothing was blocked. But since it's being blocked by crumbs or this inhibitor, the binding affinity goes down. But here's the thing, is the Vmax is unaffected. The reason is because if you have a lot of substrate, we can get back to the original velocity, no problem. So the velocity, the maximum velocity is actually not affected at all. It's just the Km. Now let's talk about uncompetitive inhibition. Uncompetitive inhibition is basically saying that this inhibitory molecule can only bind after the substrate has bound first to the enzyme. So only after the substrate has bound to the enzyme, then we have a conformational change, which causes an active site to open up for this inhibitory molecule to bind to. Now, this plot here, we can notice that the slope is the same, but the y-intercept changes. This causes the Km to be reduced, which also means a higher binding affinity, but the maximum velocity is reduced. So let's talk about this. Why? Well, the reason the binding affinity goes up is what's actually happening is when this inhibitory molecule binds, it actually stabilizes the enzyme. It doesn't break apart. So that means we need less enzyme substrate complexes around because we have so many that are actually stable. So when it's stable, actually basically the enzyme says, hey, it calls out to all the other substrates. It says, hey, come over here. I'm stable. I can handle more of you guys. Come along. So that's why I have a higher binding affinity because the enzyme is basically saying, hey, I'm stable. I can handle more substrates. That's why the Km is lower because remember, a reduced Km equals a higher binding affinity. Now the Vmax is reduced. The reason is because there's a trade-off. Even though we are more stable, we are less efficient. We are much less efficient. We still need more substrate. We need a lot more substrate to actually get to maximum velocity. Because remember, there's space being taken up by this inhibitor. It's in the way. Right? We have this inhibitor that's bound to the enzyme. That's one active site that's being taken up. Right? A substrate could take its place, but it cannot because it's in the way. Now that kind of sounds like competitive inhibition, but basically the substrate cannot bind to where this inhibitor is. But in other words, it's basically saying that this conformational change, yes, it makes it more stable, but it makes it less efficient. So it's all about efficiency. Vmax is all about efficiency. So now non-competitive inhibition is a little bit different. Non-competitive is actually very similar to uncompetitive. There are 2 scenarios. Scenario 1 is we can have exactly the same thing, is that we can have as uncompetitive. It's that the substrate binds first, then the inhibitor. Or we can have the inhibitor binds first, and then the substrate. So it's basically either or. The inhibitor can bind first, or the substrate can bind first. So non-competitive inhibition has a choice. And this causes the slope to change and the y-intercept to change. Now the Km is unaffected, but the Vmax is reduced. The reason is because, let's talk about here now. So once the inhibitor molecule binds first, it causes a conformational change. The conformational change doesn't actually make it stable. It makes it just basically the same. But what it does is it slows down the efficiency. It basically makes the enzyme a little bit less efficient. It slows everything down. That's what this basically happening. That's what non-competitive inhibition is doing. It slows things down. It doesn't affect any affinity, Nothing to do with affinity, only the speed of how fast the enzyme works. It's going to work slower now because this inhibitor, it binds first, then the substrate. So we're taking time, right? Because this inhibitor has to bind first, then the substrate. So it's taking time off, very valuable time. So that's really, basically that's it. That's the only difference here, is the inhibitor is binding first. Now, yes, I did say that non-competitive inhibition, this can also happen, where the substrate binds first and then the inhibitor, just like uncompetitive. So there's two ways, two routes this can go. It can either go this way or this way. So yes, if you found this video helpful, please like and subscribe. And until next time, later.